How are the traits we carry passed down to our children? Through the process of meiosis, which is how sex cells or gametes are made. So how does this happen? It all has to do with chromosomes, which are long and highly coiled molecules of DNA, which we will represent throughout this video with these strings of beads. So if that was a chromosome, what is this? This is also a chromosome, or rather a duplicated chromosome. It has two chromatids, which we call sister chromatids, since they are part of the same chromosome. The chromatids in this chromosome, like all sister chromatids, are attached to each other with a centromere. Sister chromatids do not have to be genetically identical to one another, as we will see in the process of meiosis. Since we have now seen multiple examples of chromosomes, you can imagine how this use of the word chromosome can be tricky, which is why it is important to know not just how the number of chromosomes change throughout meiosis, but the number of chromatids as well. Humans have 46 chromosomes, 23 from each parent, in each of their somatic cells. But for simplicity of explanation, we will be showing how the meiosis process works for a 2n=6 cell. So what does this mean? Let's take a step back. This is a haploid cell. As you can see, there is only one copy of each unique chromosome. This means that for the something n equals something equation, the coefficient of n equals 1. What about our example? It has a coefficient of 2. This means that there are two copies of each unique chromosome. When there are two copies, we call the cell diploid. You can remember the difference between diploid and haploid by noticing the prefix di, which means 2. These copies are special. Unlike when we showed one chromosome with two chromatids before, having a copy in this case means that there are two chromosomes of the same length present. These chromosomes are not identical, and thus are called homologous chromosomes. Each chromosome in a homologous pair has the same genes, so each will have the same length and centromere position. However, each chromosome in a homologous pair is inherited from a different parent, so alleles of certain genes differ between the two chromosomes. We will use this terminology later in the video, as it is very important. So now you understand the coefficient of n, but what about the number n equals? Let's go back to the haploid cell example. What can you observe? That there are 3 unique chromosomes? In case you did not already guess, that means that the number n equals is the number of unique chromosomes in a cell. For this cell, n equals 3. For our example, which is 2n=6, with a little bit of math, we can figure out that n equals 3. Thus, we have 2 pairs of 3 unique chromosomes for the 2n=6 example. Before we move on, we are going to recap what we just said. Meiosis is the process in which only sex cells or gametes are made. Chromosomes are long and highly coiled molecules of DNA. Sister chromatids are chromatids belonging to the same chromosome. Haploid cells have just one copy of each unique chromosome. Diploid cells have 2 copies, or one homologous pair, of each unique chromosome. Homologous chromosomes are identical in length, but not in function. Got all that? Great. Time to begin. The process of meiosis for a 2n=6 cell begins with 6 chromosomes, which are put into 3 sets of homologs. That weird-looking thing in the corner is known as a centrosome, and has a centriole pair, which we represented with the 2 tube-looking things inside the centrosome. Here, the nuclear envelope is still intact. Next, something called interphase occurs. Interphase occurs before meiosis 1 begins. In interphase, chromosomes in the cell duplicate, so that each chromosome is then made up of 2 identical sister chromatids. The centrosomes also duplicate. The cell is now ready to begin prophase 1. In this phase of meiosis, the nuclear envelope breaks down and homologous pairs of sister chromatids inside a cell become associated. In other words, the genes on one homolog are aligned with the corresponding genes on the other homolog. Once the homologs are aligned, the process of crossing over begins. The term crossover refers to the process in which 2 homologous non-sister chromatids exchange segments of DNA with each other. This occurs when the DNA of 2 homologous non-sister chromatids breaks at matching points. These breaks allow the broken segment of one chromatid to attach to the other chromatid, and vice versa. The points of attachment where such crossover has occurred are called chiasmata. Due to this, the sister chromatids for each chromosome are no longer genetically identical. This is what we were talking about earlier when we defined sister chromatids. Finally, As the cell prepares to move into metaphase, microtubules which stretch across the cell attach to the kinetochores of the homologous pairs of sister chromatids. Each homolog has one kinetochore attached to its centromere. For simplicity of the video, we do not show this connection until the cell has reached metaphase 1, which is the next step. Metaphase 1 involves the lining up of the homologous pairs of sister chromatids across the cell. As the homologous pairs line up, they remain attached to the kinetochores of microtubules from opposite poles of the cell. This means that the homologous pairs of sister chromatids will soon be pulled apart to opposite poles. It is important to note that the three homologous pairs can line up across the cell in a number of different ways during metaphase. For example, the pairs could line up so that two maternal chromosomes and one maternal chromosome end up on one side, while two maternal chromosomes and one paternal chromosome end up on the other side. It is also possible that the pairs line up so that all three of the maternal chromosomes end up on one side, and all three of the paternal chromosomes end up on the opposite side. Even if this occurs, the gametes will still contain both maternal and paternal DNA because of the crossover which happened previously. Once the homologous pairs of sister chromatids are lined up across the cell, with each homolog attached to a microtubule from the opposite pole of the cell, the pairs are prepared to separate. The microtubules shown here are what we mentioned earlier, as they have been attached to each homolog since the later steps in prophase 1. Metaphase 1 is complete when all of the cells are lined up on what is known as the metaphase plate. After metaphase 1 comes anaphase 1. Up until this point, proteins had been holding together the sister chromatids at the chromatids' arms. In anaphase 1, these proteins break down and the homologs separate and are guided to the opposite poles by the spindles attached to each chromosome. The next step in meiosis 1 is telophase 1, but for the sake of simplicity, we are going to group telophase 1 with a step directly after it, cytokinesis. During these steps, the cell splits into two cells and the nuclear envelope reforms around the chromosomes. At this point, each cell is composed of 3 unique chromosomes. Yes, you guessed it, meiosis 1 takes a diploid cell and produces 2 haploid cells. These 2 steps mark the end of meiosis 1. On to meiosis 2. But first, very quickly, the centrosomes in each cell are duplicated. You might be thinking to yourself, that sounds familiar. But no, the chromosomes do not duplicate as well, and this is not interphase 2. In fact, there is no second interphase step before meiosis 2. The duplication of the centrosomes is simply a necessary step for meiosis 2 to occur. Also, how we drew the nuclear envelope from meiosis 1 to meiosis 2 changed slightly, so we would like to take the time to point out that this is a nuclear envelope. Meiosis 2 follows many similar steps as in meiosis 1, so much so that they even have similar names. In meiosis 1, it was prophase 1, metaphase 1, anaphase 1, telophase 1, and cytokinesis. In meiosis 2, it is now prophase 2, metaphase 2, anaphase 2, telophase 2, and cytokinesis. Note that the label cytokinesis does not change between meiosis 1 and meiosis 2. This is because the name refers to the separating of cells, which is the same process in both. In prophase 2, spindles form and the nuclear envelope breaks down. Then the cell transitions to metaphase 2 by lining up on the metaphase plate. Note, sometimes the metaphase plate is differentiated in meiosis 1 and meiosis 2 by calling it the meiosis 1 plate and the meiosis 2 plate. We chose to keep the same wording so that it is clear that it has the same function in each case. The cell is in metaphase 2 when all of the chromosomes are lined up along the metaphase plate and attached to microtubules, similar to metaphase 1. Now we move into anaphase 2. In anaphase 1, breakdown of the proteins holding together sister chromatids at their arms occurred. In anaphase 2, the breakdown of the proteins holding the sister chromatids together occurs. This allows the chromatids to separate and be moved by the microtubules to opposite poles. Finally, telophase 2 and cytokinesis occur. The cell splits into 4 unique cells, known to the original cell as daughter cells. Each of these cells has just one set of chromosomes, meaning it is, like the cells at the end of meiosis 1, haploid. When we look at just one of these cells, we can see that the nuclear membrane reforms around the chromosomes. This marks the end of not only meiosis 2, but the entire meiosis process. So, what is this cell? If you recall, the process of meiosis generates sex cells or gametes. This means that this cell, the one you're looking at, is a gamete.